Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about immunochemistry, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. It will be focused on the 7th subsection of the specialist portfolio called immunochemistry. I will discuss knowledge requirements, competence requirements, reflective statement, evidence of achievement, Q&A suggestions, evidence ideas, and some helpful tips to make candidates' portfolio really good. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into the 7th subsection of the first section, tissue preparation techniques, called immunochemistry. The knowledge requirement explains what knowledge is expected of the candidate in relation to the relevant subsection. Candidates should be able to demonstrate their knowledge at the specialist level. Think about what knowledge they had as a Band 5 BMS. How would that differ to the knowledge of a Band 6 BMS? That specialist level of knowledge would allow the candidate to troubleshoot, problem solve, verify or validate. The first pre-analytical knowledge requirement is specific types of common fixatives and their characteristics, and their compatibility with subsequent IHC staining procedures. This requirement can be cross-referenced to the fixation subsection. Candidate needs to have the knowledge and understanding about common fixatives and their compatibility with IHC staining. You could ask the candidate to name fixatives that are commonly used in histology, to state their characteristics and their compatibility with subsequent IHC staining, to give examples of fixatives used in your lab and how they can affect IHC staining. When addressing this knowledge requirement, candidates should consider fixatives like alcohols, compound fixatives, their fixation characteristics, fixation time, antigen preservation, retrieval and sensitivity, their compatibility with subsequent IHC staining. The second pre-analytical knowledge requirement is specific types of common decalcifying agents and their characteristic, and their suitability and compatibility with subsequent IHC staining procedures. This requirement can be cross-referenced to the decalcification subsection. Candidate needs to have the knowledge and understanding about decalcifying agents and their compatibility with IHC staining. You could ask the candidate to name decalcifying agents that are commonly used in histology, to state their characteristics and their compatibility with subsequent IHC staining, to give examples of decalcifying agents used in your lab and how they can affect IHC staining, to answer if IHC staining protocols have to be adjusted for decalcified tissues, and if so, why. When addressing this knowledge requirement, candidates should consider various decalcifiers like acids and chelating agents, their characteristics, and how they affect antigenicity of the sample, compatibility with subsequent IHC staining, separate IHC staining protocols for decalcified tissue, and so on. The third pre-analytical knowledge requirement is effects on subsequent procedures with particular regard to IHC staining and the appearance of artifacts produced by poor processing. Candidate needs to have the knowledge and understanding about the effects of processing on IHC staining. You could ask the candidate to explain how processing can affect IHC staining, to name artifacts that could be seen in IHC staining if the processing is not optimal, and how to prevent or rectify those artifacts. When addressing this knowledge requirement, candidates should consider dehydration, clearing, paraffin infiltration steps of processing and their effect on antigen preservation, consequences of poor processing like antigen loss, antigen masking, variable staining intensity, nonspecific staining, background staining, compromised morphology, section lifting, and so on. The fourth pre-analytical knowledge requirement is practice and problems associated with the production of samples suitable for immunocytochemical staining from cytological specimens, to include direct smears, cytocentrifuge and liquid-based preparations, fine needle aspirates, FNAs, clots, and cell blocks. Candidate needs to have knowledge and understanding about practice and problems of various cytological samples used for IHC staining. You could ask the candidate to explain how the IHC staining of cytological specimens differ from histological specimens and why, how to stain cytological specimens such as direct smear, cytocentrifuged sample, liquid-based preparation, cell block and clots with IHC staining protocols, To name problems that could be encountered when staining cytological specimens with IHC staining and how those can be resolved. When addressing this knowledge requirement, candidates should consider sample preparation, reagents used for fixation and processing and their effect on IHC staining, antigenicity preservation, antigen retrieval techniques used, visualization, control material used, morphology preservation, cell density, possible artifacts seen, difference between histology and cytology samples and how they are stained with IHC. The fifth pre-analytical knowledge requirement is potential problems associated with prolonged section storage and loss of tissue antigenicity. This requirement can be cross-referenced to the microtomy subsection. Candidate needs to have the knowledge and understanding about problems associated with section storage and loss of antigenicity. Use the candidate to explain how the storage time of tissue section slides can affect IHC staining, and why, how the antigenicity changes over time, how your laboratory store pre-cut slides for IHC staining and why. When addressing this knowledge requirement, candidates should consider antigenicity, staining intensity, false results, variation between biomarkers, effects of oxidation, moisture, temperature and storage duration on antigens, optimal storage conditions, fixative used and so on. The sixth pre-analytical knowledge requirement is challenges associated with the use of immunocytochemistry on resin sections. Candidate needs to have the knowledge and understanding about challenges of IHC staining on resin sections. You could ask the candidate to name the challenges that can be encountered when using IHC staining on resin sections to explain how to deal with those challenges. When addressing this knowledge requirement, candidates should consider section adhesion, resin removal, antigen retrieval, antibody penetration, background staining, staining protocols, equipment needed, control material and so on. The first analytical knowledge requirement is familiarity with appropriate antigen retrieval methodologies, including proteolytic enzyme digestion, heat-mediated methods, and their mechanisms, as far as they are known. Candidate needs to have the knowledge and understanding about antigen retrieval methods and their mechanisms. You could ask the candidate antigen retrieval technique and why they are needed during IHC staining, to explain why different methods for antigen retrieval are needed, what the difference between different antigen retrieval methods is, and when would each method be used. You could ask them to explain how different antigen retrieval techniques work, and to name antigen retrieval methods used in your lab. The second analytical knowledge requirement is different proteolytic enzyme digestion methodologies, including working knowledge, awareness of the importance of optimal digestion, assessment of this in stained preparations. Candidate needs to have the knowledge and understanding about different enzyme digestion methods and their assessment. You could ask the candidate to name enzymes that can be used during proteolytic enzyme digestion, to explain why it is important to optimize digestion and what could happen if it was not optimized, to explain how the over and under digestion would look like on IHC stained slide. When addressing this knowledge requirement, candidates should consider different enzymes like proteinase K, trypsin or pepsin, their concentration, incubation time, temperature, pH and buffers used, their reproducibility, possible artifacts like tissue overdigestion, poor staining quality, high background staining, tissue variability, reproducibility, and other artifacts associated with digestion methods. The third analytical knowledge requirement is experience of different heat-mediated antigen retrieval methodologies, including working knowledge, various heat delivery systems, to include on-instrument retrieval and heated baths, and various antigen retrieval solutions. Candidate needs to have the knowledge and understanding about heat-mediated antigen retrieval techniques. You could ask the candidate to explain what heat-mediated methodologies for antigen retrieval are, to name various heat delivery systems they utilize, to explain the need for retrieval solutions. When addressing this knowledge requirement, candidates should consider microwave ovens, pressure cookers, water baths, autoclave, automated on-instrument systems, retrieval buffers like citrate buffer, EDTA, tris-EDTA, urea, and others. The fourth analytical knowledge requirement is methods for validation and verification of primary antibodies before introduction into a diagnostic procedure and the requirement for assessment of batch-to-batch variation when in use. Candidate needs to have the knowledge and understanding about validation and verification of antibodies. You could ask the candidate to explain why it is important to validate and verify new antibody before its routine use, to name steps for antibody validation and verification, to explain what batch-to-batch variation is and how would that be addressed in the lab. When addressing this knowledge requirement, candidates should consider validation and verification steps, batch variation, specificity, sensitivity, reproducibility, misinterpretation, clinical accuracy, protocol optimization, pathologist involvement and feedback, documentation, control material, polyclonal versus monoclonal. The fifth analytical knowledge requirement is concepts of sensitivity, specificity, avidity, and affinity, and their significance to the quality of immunocytochemical staining. Candidate needs to have the knowledge and understanding about antibody sensitivity, specificity, avidity, and affinity when it comes to IHC staining. You could ask the candidate to explain what sensitivity, specificity, avidity, and affinity is when it comes to IHC staining, how they can affect the quality of IHC staining, and how they influence staining protocol optimization. When addressing this knowledge requirement, candidates should consider each term, highlight how they affect quality of IHC staining, state the importance of staining protocol optimization. The sixth analytical knowledge requirement is appropriate dilution of primary antibody reagents, and the effects on subsequent immunocytochemical staining results. Candidate needs to have the knowledge and understanding about antibody dilution and their effect on staining quality. You could ask the candidate to explain, what is primary antibody? Why is dilution of primary antibody important? How does primary antibody concentration affect the IHC staining? And how to establish optimal antibody dilution. When addressing this knowledge requirement, candidates should consider antibody concentration, signal intensity, background staining, specificity, sensitivity, signal-to-noise ratio, optimization process. The seventh analytical knowledge requirement is problems of nonspecific and inappropriate staining, their causes, and methods for their reduction or elimination. Candidate needs to have the knowledge and understanding about poor IHC staining quality and how to resolve them. You could ask the candidate to name causes of nonspecific and inappropriate IHC staining, to explain how to reduce or eliminate nonspecific and inappropriate IHC staining. When addressing this knowledge requirement, candidates should consider antibody concentration and cross-reactivity, species mismatch, tissue type, staining procedure, antigen retrieval technique used, insufficient blocking and washing during staining, fixation and processing artifacts, activity of endogenous enzymes, presence of tissue during necrosis, ways to combat the problems, like use of control material, antibody titration, staining protocol optimization, robust verification and validation process, reagent quality, optimal fixation and processing of tissue, manufacturer's recommendation and so on. The eighth analytical knowledge requirement is necessity of including appropriate run controls and maintaining audit trails. Candidate needs to have the knowledge and understanding about control material and importance of audit trail. You could ask the candidate to explain why it is important to use controls during IHC staining, to name types of controls used in IHC staining, to explain how to maintain audit trail for control material, to name documentation needed when it comes to verification, use and disposal of control material, to explain how to create a control material for IHC staining. When addressing this knowledge requirement, candidates should consider positive, negative controls, agent and tissue controls, internal or endogenous controls, use of composite controls or cell lines, validation and verification, sensitivity and specificity, reproducibility, possible misinterpretation, record keeping, or traceability. The post-analytical knowledge requirement is patterns and localization in normal and abnormal cells and tissue types in order to interpret immunocytochemistry preparations for routine diagnostic use. You could cross-reference this requirement to relevant demonstration methods in Section 2. Candidate needs to have the knowledge and understanding about patterns and localization of IHC staining for interpretation purposes. You could ask the candidate to name antigen expression patterns, to explain how localization of antigens in normal and abnormal cells and tissues can differ. Why is expression pattern and localization important when it comes to IHC interpretation? When addressing this knowledge requirement, candidates should consider extracellular, nuclear, cytoplasmic and membranous expression, possible altered expression in abnormal cells, aberrant localization, heterogeneity, cell type identification, tissue specificity, distribution of positive staining, percentage of cells showing positive staining, morphology highlighted by counterstain showing exact location of positive signal, diagnostic, prognostic and therapeutic significance. Competence requirement is basically a combination of knowledge, skills, and experience candidate needs to perform a job to a satisfactory standard. Candidates' chosen evidence should demonstrate these competence requirements. The following competence requirements need to be met for this subsection. The first pre-analytical competence requirement is pre-treat slides for immunocytochemistry following local SOPs. Candidates should be able to pre-treat slides for IHC staining. Does the candidate know how to pre-treat slides for IHC staining? Do they follow relevant protocols in your lab? Do they use different protocols for FFP and fresh section? When addressing this competence requirement, candidate could consider SOPs, local protocols, slide baking, deparaffinization, rehydration, antigen retrieval, washing and blocking, reagents used during IHC staining and their function. The first analytical competence requirement is stain slides for immunocytochemistry using manual, automated methods following local SOPs. Candidate should be able to stain slides with IHC staining protocols. Can the candidate stain slides with IHC staining protocols? Are they using the correct instruments? Are they following the correct methods of staining? Are they aware of any other instruments or manual methods that can be used during IHC staining? When addressing this competence requirement, candidate could consider SOPs, steps taken to stain slides, instruments used, manual IHC staining techniques, automated IHC strainers like gap systems and flat labeling systems, multiplexing capability, advantages and disadvantages of manual and automated IHC staining. The second analytical competence requirement is optimize antibody titers, maintaining audit trails. Candidate should be able to optimize antibody titers and maintain audit trail. Can the candidate optimize antibody titers? Do they follow the correct steps during optimization? Can they interpret results of ANT titers? Can they record the process and generate relevant documentation? When addressing this competence requirement, candidate could consider validation and verification process, determining optimal dilution, specificity, and sensitivity of antibodies, reproducibility, record keeping, traceability, involvement of pathologists, control material used, result interpretation and so on. The first post-analytical competence requirement is recognize staining patterns for core repertoire antibodies on normal tissues. Candidates should be able to recognize staining patterns of core antibodies. Can the candidate recognize staining pattern of the most common antibodies used in your lab? Do they know if the staining pattern change when it becomes diseased or abnormal? When addressing this competence requirement, candidate could consider specific antibodies and their expression pattern in cells like nuclear, cytoplasmic or membranous pattern, their expression pattern change in diseased state, aberrant localization, heterogeneity, and so on. The second post-analytical competence requirement is determine the suitability of a completed immunocytochemistry run for further analysis based on included controls. Candidates should be able to determine success of IHC staining run based on control material used. Does the candidate know if the IHC staining run was successful or not? Do they know what to do if the staining was not successful? Does the candidate QC IHC stained slides? When addressing this competence requirement, candidate could consider quality control of stained slides, control material used like positive and negative controls showing expected results, staining intensity, possible issues like ground staining, no staining within positive control, abnormal staining within negative control, non-specific staining and so on. The third post-analytical competence requirement is analyze the causes of suboptimal immunostaining. Candidate should be able to troubleshoot suboptimal IHC staining. Can the candidate resolve issues with suboptimal IHC staining? Do they take correct steps during troubleshooting? Do they correctly record actions taken? When addressing this competence requirement, candidate could consider weak or absent staining and possible causes like overfixation, or autolysis due to delayed fixation, background and nonspecific staining and possible causes like poor blocking or washing steps, gradient of staining quality caused by poor penetration of reagents due to tissue thickness, perfections, record keeping and so on. Each section also contains reflective statement which is asking the candidate to summarize their role in the lab in the context with each section. When they complete all the subsections within each section, they will be required to write a reflective statement. They also need to write a personal reflection on their training and why they chose their evidence. To prepare for that, ask them to write a smaller reflective statement for each subsection. What is their role in context of this subsection within the lab? What does their evidence demonstrate? Ask them to write a smaller justification as to why they chose their evidence for this subsection. Also ask them to write about what was their training like when to this subsection. Writing a smaller reflective statement at the end of each subsection will then make it much easier for them at the end of the section. Each subsection also has evidence of achievement. This is a part where you, the training officer, sign the candidate off as competent and fit to practice as a BMS at the specialist level. By doing that, you will confirm that the candidate can work in accordance with local SOPs relevant to this subsection. This will also confirm that candidate has answered questions set by you to test their knowledge and skills relevant to this subsection. And you will also confirm that candidate's chosen evidence demonstrates their competence relevant to this subsection. The candidate will need to be signed off as competent by an internal assessor for each subsection. They can be signed off by you, the training officer, senior BMS overseeing relevant section, or by the training lead. These are some example questions you could give your candidate to test their knowledge and skills about this subsection. These should be based around the knowledge and skill requirements mentioned earlier. These are some more example questions you can ask your candidate to complete, to test their knowledge and skills about this subsection. You could ask the candidate to complete tables you created, based on knowledge requirements. Supporting evidence should be selected by the candidate. It needs to be based around the competency requirements, and be relevant for each subsection. The evidence should allow the candidate to demonstrate their competence in each subsection. It should be specific and relevant to the candidate's in-house practice. It should also demonstrate candidate's specialist competence. If you would like to see specific ideas for supportive evidence, please have a look at the video for trainees. Link will be included in the video description. I would also like to share with you some helpful tips that can make candidates' portfolio look really good. Ask them to try and present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs or even videos, if they can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Ask them to try and make it a little bit more interesting to the reader, which will be you, and eventually the assessor. Ask them to try and put themselves in your or the assessor's shoes. How would they like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture 1,000 words. I will make a video where I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also ask them to try and use various types of evidence, not just one type, like case studies, for each subsection. They could use experiments, case studies, annotated results, annotated photographs of them performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation verification evidence, comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Ask them to try and bring in their personality. Ask them to think about the ways they like to learn best. If experimenting, let them perform experiments. If they like troubleshooting, let them perform troubleshooting. Ask them to choose something that demonstrates their specialist understanding, knowledge, and skills, and how they apply this in their practice. And one more thing, ask them to keep the competence requirements in mind when they are selecting their evidence. Good evidence will tick off several requirements. And this brings me to the end of today's video. Hopefully you now have a better understanding about the knowledge and competence requirements for this subsection. And I hope you also have a better idea what questions you can choose to test candidates' knowledge. And my final take-home message? Keep those requirements in mind when selecting questions and guiding the candidate. Bye for now.